participating in this webinar today, and in particular to Selen and Naomi, who put all of this together, together with Romy, and thank you all three. So I think we're going to have a very useful morning, and I'm going to start by introducing the RAPSODI and Frontline studies by handing over to Selen and to Naomi to discuss those. But let me first of all tell you a little about these two researchers. So Selen has a BSc in Biomedical Sciences, an MSc in Biomedical and Molecular Research, and coordinates the PD Frontline study. She's interested in research that underlines early determinants of Parkinson's disease to develop strategies for medicine that is preventative, precise, and clinically translational. Naomi Limbachiya has a BSc in Psychology, an MSc in Neuroscience, and a postgraduate diploma in Genomic Medicine, and coordinates the RAPSODI study. Her interests lie within the early identification of Parkinson's disease, including genetic, clinical, environmental, and epidemiological factors, again, to innovate effective therapeutics that can improve global health care for those with Parkinson's disease, and to contribute to health equality. So, Selen and Naomi, over to you. Thank you very much.
Thank you, Professor Schapira. So we're going to begin by talking about the foundation of both the RAPSODI and PD Frontline study, and this would be the GBA gene. So we each inherit 2 copies of every gene from each parent. People with Gaucher's disease, which is a lysosomal storage disorder, have a genetic alteration in both copies of the GBA gene. People with a genetic alteration in one copy of the GBA gene will not develop Gaucher's disease, but they do have a slight increased risk of developing Parkinson's disease. This is still a very low risk, and most people will not go on to develop Parkinson's disease.
We are now going to go over briefly the connection between GCase and Parkinson's disease to explain why this is relevant to our studies. So, as a bit of background, GCase is the enzyme that is made by the GBA gene. An enzyme is a protein that mediates certain reactions, and the GBA gene makes the protein glucose cerebrosidase, or GCase. So, in GBA gene where there isn't a fault, you have the breakdown of alpha-synuclein. Alpha-synuclein is a protein that builds up in neurodegenerative conditions such as Parkinson's disease and causes neuronal cell death. So in general, when GCase is low, alpha-synuclein builds up. This is something that we often see in people with Parkinson's disease. And when there is a fault in the GBA gene, this leads to the alpha-synuclein clumping together, and this causes the neuronal cell death in Parkinson's disease where there is a fault in the GBA gene.
I will now go into to discuss the RAPSODI study with you. And our mission is to slow down and prevent the onset of Parkinson's disease. In terms of the purpose of the RAPSODI study, we want to understand why there is this risk of developing Parkinson's disease in some people, but not in others. And this is particularly salient in communities where this faulty gene is more common. So an example would be people of Ashkenazi Jewish heritage who have a slight increased risk of being a GBA carrier. But it's quite important to note that this faulty GBA gene can be found in people of all backgrounds. So the purpose of the RAPSODI study is really to define what the early signs of Parkinson's disease are in people with a faulty GBA gene, so that when there are treatments to prevent Parkinson's disease, we will know who will benefit from these and who we can offer them to. What does the study involve? The RAPSODI study is a nationwide online study involving yearly assessments, and each assessment takes approximately 45 to 60 minutes to complete. There's a total of 6 assessments to complete. In terms of what we actually measure, we measure a range of symptoms, including mood, sleep, movement, attention and memory, sense of smell, and DNA. I think it's quite important to know that we understand the sensitivity that comes with genetic results, and we understand that it's a personal choice. And so not everyone will want to know their genetic results. So the RAPSODI study actually gives all our participants a choice as to whether they want to know and as to whether their GP or clinician also is to be informed. And if participants do want to know their genetic results, they're given free genetic counselling with a member of our team. And this includes a member of our team discussing the change that we found within the GBA gene and what this means for them and the potential implications this may have for their family members. So who can actually participate in the RAPSODI study? GBA carriers, so this is individuals who carry one faulty GBA gene, people with Gaucher's disease, relatives of people with Gaucher's disease, relatives of people with Parkinson's disease, people with Parkinson's disease who also carry one faulty GBA gene, and all our participants must be over the age of 18 and a resident in the UK. So signing up is relatively easy. All you have to do is visit our website and we'll ask you to provide a few details about yourself, such as your name, your date of birth, your email address. Once your account is created, you will automatically be able to see our participant information sheet and a consent form that pertains to this. And once you've signed this consent form, you're officially part of the RAPSODI study and you can begin your first assessment. So just to give you an overview of the RAPSODI study, we're trying to measure symptoms of Parkinson's disease in carriers and non-carriers of the GBA gene. We then want to study the similarities and differences between those who develop Parkinson's disease and those who do not. And then we want to use this information to develop treatments that can actually prevent and slow down the progression of Parkinson's disease. I will now hand over to my colleague Selen, who will discuss the PD Frontline study with you.
Thank you very much, Naomi. Sorry. Screen. I don't seem to be able to share my screen. I'm not entirely sure why.
Would you like me to show your slides?
Perhaps that may be the best way. Yes.
Absolutely.
Thank you, Naomi. So I am the study coordinator for PD Frontline, and I'm going to just go over the purpose of the study and a little bit of background as to the purpose as well. So next slide, please. So the purpose of PD Frontline, there are four main purposes of PD Frontline: to genotype, to act as a registry, to refer to clinical trials, and a small element of the family tree. So the risk factor genes GBA and LRRK2. So as we mentioned, the GBA gene is a risk factor for Parkinson's disease. And as Parkinson's researchers, that's a very exciting juncture where for the first time, research which can be targeted towards these genes, which could prevent or slow down the progression of Parkinson's, is really a possibility. So what we wanted to do was to have a platform for people with Parkinson's disease who wanted to be registered to a platform which would genotype them and make them and allow them to be put forward for clinical trials and studies which target these genes. And not only these genes, but just other studies which could help our understanding of Parkinson's disease. So the whole purpose of PD Frontline is developing a cohort of genotyped individuals who can be referred onto future trials. And then in terms of the family tree element, we do ask some questions about the familial history of Parkinson's disease within your lineage to help better understand how Parkinson's disease can be passed on. So what does the study involve? So currently we have over 1,860 participants, and we recruit just all across the UK, and who can take part. So with PD Frontline, unlike RAPSODI, we only recruit those who are diagnosed with Parkinson's disease and do not know that they carry the GBA gene. Signing up to PD Frontline is very, very easy, very, very straightforward. So this is what our homepage looks like, and you would click on Register for PD Frontline. So once you do this, there'll be some qualifying questions which you're required to answer. You must live in the UK, you must have a confirmed diagnosis of Parkinson's disease, and you must not know whether you carry the GBA gene. If you do know that you carry the GBA gene, you can sign up to RAPSODI instead. You then complete the registration details, which just ask questions like your name, your date of birth, ask you to set up a password, and your email address. Once you verify your email, you'll be able to complete our consent form, which gives us permission to send your results to your GP or clinician and how to generally handle your data, as well as giving you the option to read our participant information sheet. And then you complete our short study survey. The whole process should take between 10 to 15 minutes from start to finish to complete. So once you've completed the sign up and you've completed the study survey, we will then send you a saliva kit, asking you to provide a sample of saliva to send back to us in a prepaid envelope. This is usually sent out within 2 to 4 weeks of you completing your study survey. Your sample is then sent off for genotyping and analysis, and then we inform you of your results. So unlike the RAPSODI study, participants of PD Frontline will automatically be informed of their results. But you do have the option to inform or not inform your GP and your clinician. So now Naomi and I are going to present a little bit about how our studies are connected and some common themes. So we thought it would be very helpful to give you a timeline from start to finish, from you signing up and your results actually being ready. So the first part, when you sign up and you receive a saliva kit, as I mentioned, takes about 2 to 4 weeks. After this, the genotyping process takes about 14 to 16 weeks. The genotyping process has 2 steps to it: initial novel MinION sequencing, followed by Sanger confirmatory sequencing. This is to ensure that all our results are NHS confirmed, and the results are also analysed by our geneticist. We then, Naomi and I then process the results, which takes about 4 to 6 weeks for us to ensure that all the results are correct and we can inform everyone individually, providing genetic counselling where necessary. We also wanted to explain how our studies are connected. Some of you may already have experienced the reassignment process, but we thought it would be very, very helpful to give you a summary and overview of our studies and how they are actually connected. So as I mentioned, PD Frontline, those who have idiopathic Parkinson's disease, so they do not have an underlying cause of the Parkinson's disease with an unknown GBA status, they register PD Frontline. They complete the registration, they are genotyped. If they're GBA negative, they remain in PD Frontline. If they're GBA positive, they're referred to our sister study, RAPSODI. And I would also like to note that both RAPSODI and PD Frontline participants are offered the exact same clinical trial and observational study opportunities. I'm going to hand over to Naomi.
Yes, thank you, Selen. So once you're referred to the RAPSODI portal, your account is already created, so you won't have to complete any of that again, but you will be sent a smell test to complete. And along with this, you can complete our online assessment. So we're going to be measuring the same things I mentioned earlier: your mood, your sleep, and your attention and memory. And yes, you'll officially be part of the RAPSODI study. So in terms of, as Selen mentioned, both PD Frontline and RAPSODI participants are given the same opportunities. So an example would be the phase 2 Ambroxol drug trial, which was offered to both RAPSODI and PD Frontline participants. And a very exciting project that we are currently running is the PD Biome project, which is investigating the link between the gut microbiome and the brain. And Dr. Menozzi will give you some more information about that. And we actually have a very, very exciting upcoming study, clinical trial, that Professor Schapira will give you some more details about. And we also want to thank our participants who are always giving us feedback. And they express that they're really enjoying the study, which is very important to us, and that it's convenient, and that they can also get their family members involved, which is something that they really enjoy. So we're very happy to hear that. And yes, there are many ways that you can get in contact with us. So you can email the RAPSODI or PD Frontline team. You can also call us. You can directly visit our website and sign up, or you can read some of our tweets on our Twitter profile page.
Before we hand over to our panelists, I just wanted to mention that you can put any questions you have in the Q&A section, and we'll address everything at the end. So as we go along, any questions you have directed to specific panelists, please do put that in the Q&A. Thank you.
So yes, we will now hand over to our panelists, who are all well-renowned experts and leaders in the field of Parkinson's and Gaucher's disease. Our first speaker is going to be Professor Derralynn Hughes, who is a Professor of Experimental Haematology at the University College London. She is a Clinical Director of Research and Innovation at the Royal Free London NHS Foundation Trust and Co-Clinical Director of the NCL Cancer Alliance. She is also Chair of the International Gaucher Alliance. Professor Hughes is a pioneer in Gaucher's disease research, and she will now present the link between Gaucher's and Parkinson's disease.
Thanks very much, and thanks for the invitation to talk this morning. I hope that you can see my slides okay. Yeah. So I'm going to talk a little bit about Gaucher disease, just to put it in context, and then some thoughts on the link between Gaucher and Parkinson's. So these are my disclosures. Okay. So Gaucher disease is a rare inherited metabolic condition. It affects probably around 1 in 40,000 of the population overall, but more so in certain communities. So it's very common, for example, in the Ashkenazi population. And it results from the deficiency of the enzyme that you just heard described, glucocerebrosidase. And the normal function of that enzyme is to break down a material called glucosyl ceramide. So it's a sugar with ceramide. And you'll hear talk about this sometimes, for example, when people are describing hair products and so on. They talk about ceramides, and it's because it's a very important molecule that's involved in skin and hair and blood cells and other components of the body. However, in the normal functioning of the body, we have to break down glucosyl ceramide and then recycle it. So if that enzyme, glucocerebrosidase, is missing, then instead of it being broken down to its constituent parts, it accumulates in various cells in the body. So it can't be reused, but also its accumulation can cause problems. And it accumulates particularly in a sort of cell called a macrophage. And you've got a picture here of some macrophages within the bone marrow. And these are normally fairly small cells, but here they're much enlarged and they are really full of the glucosyl ceramide. So these are very distinctive cells. And it's these cells, amongst some of the other disturbances in the body, that cause problems with Gaucher disease. Now, the difference between Gaucher disease and what you've just been hearing about is to inherit Gaucher, you need to have abnormalities on both of the GBA genes. So we've got a gene from each parent, and to be a carrier, you would have an abnormality on one of those. But to be a patient or a person with Gaucher disease, then you would have abnormalities on both of them. And that can cause a number of problems in the body. For the most part, patients present with a large spleen, that's an organ in the abdomen, or a large liver, and that causes problems with the blood cells. So the platelet cells are low, and so people can have bruising, bleeding, bone pain. There are problems with the bones that lead to fractures, and also problems with energy levels, for example. So the symptoms are up here at the top on the right-hand side. So fatigue, bruising and bleeding, heavy periods in women, decreased appetite, and abdominal pain. And these are the most common features of Gaucher disease. This is some work that we've been doing in the lab just for your interest to show how those cells, which are Gaucher macrophages, can become enlarged and abnormally functional. And I think this is quite a nice picture. This is an electron microscopic picture of some bone on the right-hand side. And in the very right are cells from a normal person, from a control individual. And you can see very little tiny pits, whereas those on the left-hand side are big pits carved out of the bone by those Gaucher macrophages, showing that they're very, very active and cause lots of problems around the body. And you can see here the size of the cell in the panel on the left-hand side, much bigger than the ones from people who don't have Gaucher disease. However, we've come to realise over the last 10-15 years or so that there are some less common manifestations in Gaucher disease, but they're very important. They may not affect quite as many people, but when they do, it can cause significant issues for them. And one of those is Parkinson's disease. There are some other less common manifestations which are also difficult to understand and may relate to other factors, for the increased incidence of some blood cancers, for example, myeloma. So we first came to understand this some years ago now, where a researcher in New York started to look at the numbers of patients in his population who had various problems with bone. And so there was a survey of 55 patients with Gaucher disease type 1. This is the type of Gaucher disease that is most common and affects adults and is not typically known to have manifestations that affect the brain. What he found is that we would, as expected, some of those patients had problems with the spleen and with the bones, but 6% of those, so 6 out of 100, had Parkinson's disease. Further work has shown an increased incidence of Parkinson's disease amongst patients with Gaucher disease, as well as amongst carriers who have at least one GBA mutation. So almost a third of patients with Gaucher disease reported to have a relative with Parkinson's disease, and some patients themselves were found to have the condition. So this has led on to work over the years trying to understand how, if we put all of the patients and their relatives together across many different treatment centres, what it would tell us about the relationship between the GBA gene and Parkinson's disease. And this work is now 10 or more years old, but it was the first time really that we had a lot of data which suggested that the GBA gene was a common risk factor for Parkinson's disease, both in carriers and then also in patients themselves. And in fact, in Gaucher disease type 1 patients, the risk of having Parkinson's is probably about 20 times that in people who don't have a abnormal GBA gene at all. There are some similarities between the mechanisms of Parkinson's disease in Gaucher patients and carriers, and this has been investigated both by understanding the sort of physical changes that people have, but also understanding some of the changes that people have in their brains on imaging studies. And this is the sort of investigations that are ongoing to really try and understand what's happening with the GBA gene that makes it more likely that people have Parkinson's disease. So this work tries to show what the natural history is of Parkinson's in Gaucher disease. So what are the symptoms that people first experience, and how does that relate to the things that we can measure in the blood, and then how does that progress over time? So the early phase symptoms, which seems to be going on over the first 10 years or so, are things like loss of sense of smell, abnormal sleep patterns, depression and anxiety, constipation and problems with memory and thinking. And those are associated with low levels of the enzyme, which is what is coded by the GBA gene, some differences in the substrate material, and differences in the imaging, which I just showed to you. And then over time, we see that that changes into what is known as a clinical phase, where people might actually have more recognisable problems at a younger age, perhaps, than in the general population, and with more emphasis on memory and thinking problems, as well as the movement problems that people have. And so this gives us a really important insight into mechanisms in Parkinson's disease that might relate to GBA, but also to Parkinson's in general. These are the most common molecules that we talk about in both Gaucher and Parkinson's. So the glucocerebrosidase enzyme, the molecule that accumulates, the glucosylceramide, and the synuclein molecule that's important in Parkinson's. Now, I'm not the best person in this panel to be talking about the possible mechanisms between Gaucher and Parkinson's disease, because you have a lot of many years of research looking at that. But it would seem that there are important links between the alpha-synuclein molecule, the glucocerebrosidase enzyme, and the glucosylceramide, which interact with each other and appear to promote more aggregation of the alpha-synuclein, leading to a greater likelihood of Parkinson's disease. This has led us to really reevaluate how we classify Gaucher disease. So I've talked about type 1 disease. This is the disease that affects older children and adults. Mostly has not been thought to have severe brain or neurological manifestations. Type 3 occurs in young adults and has more chronic neurological manifestations, including problems and seizures. And type 2 affects very young children, and usually it has a very rapid and unfortunate outcome in those small children. But what we've begun to think is actually there is more of a continuum, and this includes those Parkinsonian manifestations in patients with type 1 and type 3 disease. So what can we do about any of this? Well, there are many now specific therapies for Gaucher disease which are aimed at reducing the amount of substrate, the amount of that glucosylceramide which accumulates in Gaucher disease. And these originally were things like bone marrow transplantation, and then giving the enzyme back by intravenous infusions. And then over the years, we've developed oral treatments which privation of substrate, which prevent that substrate being made rather than just breaking it down. And those have now new formulations as well. So it's going to be important to look to see whether any of these types of treatments which address Gaucher disease might also be helpful in addressing some of the problems or some of the association with Parkinson's disease. It's unlikely that enzyme therapy will be helpful because the enzyme that we give into the vein doesn't get through into the brain. However, some of the oral treatments might be able to penetrate the brain, so it's going to be important to look at the effects of those. And then just my final thought is that really, from the point of view of rare disorders and from Gaucher disease, we are progressing very rapidly to thinking about gene therapy. And in fact, we now have a gene therapy study open in our department in the hospital as a potential treatment for Gaucher disease, looking at how it affects Gaucher disease. And this is either infusing a gene therapy via a virus into patients, or in some cases by stem cells. They work in different ways, and I can describe that in some other context for you. But it might be that different mechanisms of introducing gene therapy, particularly if it's directed towards the brain, might also be important in the long term of treatment for some of the neurological manifestations of Gaucher disease. So there are a number of things for us to think about. Which are the Gaucher patients? Why are carriers at risk, and how can we make the diagnosis, and how can we make the treatment? So these are my conclusions, but I'm very happy to take any questions either now or in the chat or in a discussion later. So thank you for your attention, and I will stop sharing.
Thank you very much, Derralynn. Yeah, I think we'll do the Q&A at the end and direct any questions they have for you then. Next, we have Helen Matthews. She is the Deputy CEO of Cure Parkinson. She has been involved with the charity since its inception and worked alongside the charity co-founder, Tom Isaacs, since 2002. Helen specializes in PR, marketing, event planning, and administration. Helen oversees the charity's current research and patient initiative projects, as well as the day-to-day operations of the charity. Helen sits on Cure Parkinson's international linked trials, clinical trials, and research committees. Over to you, Helen.
And I failed on the first technical hurdle, which was turning off my microphone. Thank you so much for having me with you this morning. It's lovely to be here, and it's lovely to have a chance to tell you a little bit about the work that we do at Cure Parkinson's, but also I think to try and put into context why PD Frontline and RAPSODI are critical studies to help involve people with Parkinson's in research that's in the pipeline. So I'm going to very much be looking at the pipeline of trials that are coming and why these projects are so relevant to you. So I wonder, I don't know who's in charge of my slides, but thank you very much. That's really helpful. So just a little bit about Cure Parkinson's if you haven't come across us before. We were founded by 4 people living with Parkinson's, one of whom was Tom Isaacs that Selen mentioned, who were passionate to drive research into a cure for Parkinson's. And so we involve people with Parkinson's in every step of our work and our research. For us, cure means changing the course of Parkinson's by preventing it, slowing it down, stopping it, and even reversing it. We only fund research that has the potential to do this, and we fund preclinical research as well as clinical trials. We want to find solutions for all sorts of Parkinson's. Could we go on to the next slide? So one of our key initiatives that we have, which was set up actually 10 years ago, is our International Linked Clinical Trials Initiative. In essence, we scan all the research literature every year to try and understand what drugs are coming through that might be relevant in Parkinson's. We discuss with biotechs their drugs that might be of interest. We talk to big pharmaceutical companies as well, but we also talk to investigators who are working on old drugs that have been around for ages and been used to treat other diseases, to really explore the work that they are doing with those as to whether they might be relevant for Parkinson's. From that, we develop a long list of potential drugs or potential agents to slow down, stop, or reverse Parkinson's. Then we really go into the detailed analysis of each of those drugs to try and understand their potential benefit for treating Parkinson's. We look at how they work. We look at the strength of the preclinical data in the different types of models that we can use. We look at clinical data, any clinical data in any disease area as to how it might be relevant. But critically, we look at safety, and we also look at mode of administration of that drug to see whether it's something that could be doable for people living with Parkinson's. From that, we develop a short list of drugs. We then generate really detailed dossiers. And by detailed dossiers, I mean, 20 to 30 pages of detailed analysis of what this drug and its potential in Parkinson's. So really exploring all the evidence that we have, but then presenting it in a way that to try and create a compelling case as why it might be relevant to move forward in Parkinson's. From that, 15 to 20 of these dossiers are reviewed by our international panel of researchers who are clinical specialists, preclinical specialists, clinical trial specialists, drug discovery experts, and they debate these dossiers over a 2-day meeting. And then the top ranked of those, we try and prioritize to move forward into clinical trial. Could we move on to the next slide, please? So in 10 years, what have we done? We've completed 13 trials involving 9 drugs, which has involved 1,324 people currently. Ongoing, there are another 13 trials of ILCT prioritized drugs, and that's involving another 2,500 people with Parkinson's. And in the pipeline, bizarrely, I did the numbers not very long ago. It is 13, 13, and 13. We're currently sitting with 13 drugs that are prioritized for trial, 10 of which are highly prioritized by our committee. We don't fund all of these by ourselves. We believe very strongly in working in partnership. And so we have a co-funding agreement with the Van Andel Research Institute, who we work very closely with in this program. And for example, we are co-funding with them some sub-studies in the exenatide phase 3 trial that's ongoing. We've been co-funding with them on lixisenatide and liraglutide trials. Lixisenatide has been taking place in France and we'll have some results later this year. And the liraglutide trial has been happening in Cedars-Sinai in the States. Nilotinib, we also co-funded, but that was also with the Michael J. Fox Foundation, and that took place in the US. And we've also been working on Ambroxol, which we've co-funded with the Van Andel Institute, the Ambroxol phase 2 trial, and we're working with them to package the funding for the phase 3. So this gives you a sense of the pipeline as it stands at the moment. So some of the drugs I've just been talking about, you can see that lixisenatide and liraglutide, UDCA, NAC, and deferriprone are all in blue. That means that we've got results that will be coming through a little bit later this year on each of these drugs, each of which has been prioritized by our ILCT committee, some of which we have funded directly, including UDCA, some of which have been picked up by other funders. Exenatide right at the top. This is a whole program of research that we have been working on since actually 2009, when we funded the very first study. Then Michael J. Fox funded the phase 2 study. And then the phase 3 study has been predominantly funded by the UK government, but with support from Cure Parkinson's. Ambroxol, we funded the phase 2 study, and we're working towards phase 3 for that. Deferriprone, this removes the iron buildup in the brain. And again, this has been a pan-European study, and we will expect some results from that a bit later on this year. But just going back to the exenatide right at the very top of the list, there are a number of drugs that are in the same class of drug, including lixisenatide, liraglutide, and NLY01. Yes, it is that way around. I managed to get the acronym the wrong way around the other day, and I was very annoyed with myself. Anyway, all of these are they're a type of diabetes drug that could have real importance in Parkinson's, and we're very keen to see this story move forward. But each of those trials is answering a slightly different question about by studying a different group of people living with Parkinson's, or by involving people, for example, with insulin resistance and Parkinson's. So we're trying to get more of a picture with each of these trials. Nicotinamide riboside, that's taking place in Norway. That's a trial that works on the energy of the cells, rather like UDCA does. K0706, this is a drug that is used in cancer. Then we've also got fasudil, nortriptyline, and azathioprine, all in trials. Nortriptyline in the UK, azathioprine in the UK, fasudil in Germany, actually. And then in the middle, you can see the Australian Parkinson's Mission. They have got, they're testing 3 drugs against placebo in the first trial, and they're just in the throes of planning their second trial. So you can see it's a very rich environment of trials that are ongoing. And actually, if we could just go on to the next slide. And very annoyingly, on Friday, and I should have updated it, there was a new paper that came out that looks at the pipeline of clinical trials. This has been predominantly written by Kevin McFarthing, who lives with Parkinson's and is a chemist by background, who works very, very closely with Simon Stott, who's our Deputy Director of Research and Development. And actually, there's a line across the middle of that circle. It's a very, 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 very busy picture. But if you look above the circle, the top half of the circle, those are all the drugs that have the potential to be changing the course of Parkinson's. The outer circle is phase 1, so that means they're in their very, very earliest stage of testing. The next circle in, that means it's in phase 2. That means the trials are taking place in people with Parkinson's. And there's a real mixture of drugs in there, a number of which you'll have seen from my previous slide. Then in phase 3, you can see last year there were only 2 drugs that were in phase 3 that had the potential to be changing Parkinson's, slowing it down, stopping it, or reversing it. You can see the second half of the lower half of the diagram is really cluttered. Those are all drugs that are being used to treat the symptoms of Parkinson's. So the top half is about changing the disease progression. The lower half is about managing the symptoms of Parkinson's. So it's really important that we keep the focus on that, because you can see in the central circle, which is the phase 3, which is the final section of clinical testing, the number of drugs that are being developed to manage symptoms, but the very few that are being progressed at this stage to actually try and change the course of Parkinson's, to try and slow it down, to stop it or reverse it. So just it gives you a snapshot of the amount of energy that's going on in Parkinson's research, but also actually how predominantly there's more effort in the symptomatic treatment than there is in the managing and trying to slow the progression of Parkinson's, but that also it's a rich pipeline. We've just got to try and keep moving things forward into phase 3. Could we go on to the last of my slides? So this is actually of particular relevance to PD Frontline and also to the RAPSODI studies, because as you've heard, what we're trying to do is to identify people who do carry genetic risk factors for Parkinson's or have Parkinson's and these genetic risk factors. And these are trials that are in the pipeline for these forms of Parkinson's. So you can see the first one on the list is a Biogen and Denali collaboration, looking at LRRK2 inhibition. And this is going into phase 2, but it's also will be starting a phase 3 trial later this year. And they will be recruiting participants from the UK as well as across the US. BIA, yes, so this is a really interesting drug. This is particularly for GBA Parkinson's. And again, I think that they will be moving into phase 2 trials in 2022. Not quite sure of where yet. It might be in the Netherlands, maybe even New Zealand, but that's a very interesting drug. So particularly for GBA Parkinson's. Another one for LRRK2 is in its early phase testing in the US. Escape Bio, but again, they have huge ambitions to move this forward and move this forward rapidly. They also have a parallel drug that they're moving forward for GBA, which will enter phase 2 trials probably in 2023. Vanquo Bio, this is another drug that is being developed for GBA, and that's in its preclinical phase. And another one that's also in preclinical is being developed. I don't know who's behind it. That one, actually, the last one. But again, it gives you a sense that there is a pipeline that is absolutely coming and is relevant for this particular form of Parkinson's, these particular forms of Parkinson's. So I hope that that gives you a sense that, you know, you taking part now is really so helpful for the research field for the future, but it is also putting you in a place that these opportunities may be relevant for you in the very near future. And I think that's me done, and I hope I haven't gone over time.
Thank you so much, Helen. That was excellent. I will now be introducing our next speaker, who is Dr. Laura Smith. Dr. Smith is a research associate at the University College London. Dr. Smith's research focuses upon the cellular and molecular mechanisms underlying Parkinson's disease. Dr. Smith will now present the topic of the enigma of the E326K GBA gene in Parkinson's disease.
Thank you, Naomi. Yes, I'm going to talk about the E326K GBA gene variant because that's the mutation that I study in the lab. So just a bit of background, I think Derralynn covered it quite well, but the GBA gene encodes for the GCase enzyme, which breaks down these fats here into their products, and it's these fats that accumulate in Gaucher disease. So for the purpose of this talk, I'll refer to these as Gaucher disease fats. So just some background of what happens in the cell. When you have the GBA gene, it is encoded in the nucleus and it creates the GCase GBA enzyme. And when it has no mutations, this enzyme is folded correctly and it will be shuttled through something called the endoplasmic reticulum and trafficked to the lysosome. And at the lysosome, the GBA enzyme can do its activity, it can help with the lysosome's function of breaking down the Gaucher disease fats and breaking down other proteins such as the alpha-synuclein protein, which is the toxic protein that builds up in Parkinson's disease. The GBA enzyme may also have a role in the cell with helping with the function of the mitochondria, which is the part of the cell which is responsible for making energy in the cell. And it does this by using things such as sugars and other fats such as fatty acids to provide fuel to generate energy. So when you have a mutation in the GBA gene, the normally folded GBA protein can become misfolded. This can cause the protein itself to have a reduction in activity, or it can cause the protein to be stuck inside the endoplasmic reticulum. And then this can cause it to activate some toxic pathways associated with the endoplasmic reticulum, which can be toxic to the cell. Both of these will mean that either less GCase protein is getting to the lysosome, or if the protein is getting to the lysosome, but it's misfolded, it isn't active. And this can reduce overall lysosomal function. And this can lead to the buildup of these Gaucher disease fats and also the buildup of other proteins such as alpha-synuclein. It may also lead to a dysfunction of the mitochondria, and this may be potentially to do with altered breakdown of things like sugars and fatty acids and energy production. So my work focuses on the E326K mutation. It's a very interesting mutation because as the previous talks have said, when you have two photocopies of the GBA gene, you get Gaucher disease. However, when you have two E326K copies, you do not develop Gaucher disease, but it is still associated with an increased risk of Parkinson's disease. So it's referred to as a risk variant. Other Gaucher disease causing mutations can normally be stratified into either severe or mild mutations, depending on how they present in Gaucher disease, depending on how they affect the activity, and then depending on how the type of Parkinson's that they lead to, whether this is severe, whether there's a higher risk. So the two most common GD causing mutations are L204P and N370S. So L204P is generally a severe mutation and N370S is generally a mild mutation. I just put this table just so you can compare the E326K to the GD causing mutations. So for my work, I wanted to figure out kind of what's going on with the E326K mutation inside the cell. And to do this, I set myself 4 questions that I want to answer. Does the mutation reduce GCase function? Does it cause the protein to be folded incorrectly and stuck inside the endoplasmic reticulum to activate the associated stress pathways? Does it lead to this buildup of toxic alpha-synuclein aggregates? And does it affect the level of fats in cells? So to do this, I use human cells to look at the way that the protein behaves inside of them. And a lot of my work is done on skin cells. So when you come in for visits and the doctor will take a biopsy, That then gets given to us in the lab, and we can isolate your skin cells away from fat cells, and then we grow your skin cells in the lab, and then use these to figure out what's happening with the GBA protein inside of them. We also use brain cancer cells, neuroblastoma cell lines. These are used quite a lot in Parkinson's disease research because they're quite neuronal-like. But if we want to make them more neuronal, we can add chemicals to them and we can transform them into neurons. So this is what I would see if I look under the microscope. I think it's quite cool because you can see all the different cell bodies, and all these long projections are the different nerves connecting to each other. And then here is just when we stain it. So the blue is each different cell, and then the green is the long axons of the neuron. So first of all, I have looked to see if the E326K GBA gene affects GBA function in human cells. So first of all, we can measure the protein levels in the cell, and we do this by doing something called a Western blot. And the bands here represent the amount of GCase protein in the sample. And you can see that the control is pretty similar to E326K. And when we quantify it, we do see that in the E326K cells, there's not much reduction in protein level compared to control, which is very different than what we see in the other two common Gaucher disease causing mutations. We can also measure the activity in the cell, and we do this by adding an artificial substrate to the cells. And then the GCase that's active in the cells will break down this substrate, and the product that you get is fluorescent. We can then measure the fluorescence, and that gives an indication of the activity in the cells. And again, you can see that with the E326K, there isn't a reduction in activity. So from this, we can conclude that in the skin cells that we look at and the neuroblastoma cells, there isn't a reduction of GBA function in those with E326K. So then I turned my attention to looking to see if the mutation causes misfolding of the protein. Does it cause it to be stuck in the endoplasmic reticulum and activate toxic pathways associated with that? So to do this, we can measure the amount of GBA protein that is immature and hasn't been processed fully, which means that it's likely to be stuck in the ER, in the endoplasmic reticulum. And in the E326K cells, there isn't an increase in misfolded protein that's stuck in the endoplasmic reticulum. We can also measure the level of two proteins. One is called BIP and one is called CHOP. And these two proteins are increased when you get the activation of these endoplasmic reticulum stress pathways. And again, the E326K doesn't seem to be activating these pathways. It's pretty similar to control. So it's likely that this mutation doesn't cause the protein to be misfolded and stuck in the endoplasmic reticulum. So then I wanted to see, is this mutation even associated with alpha-synuclein aggregation that we normally see in Parkinson's? So normally, alpha-synuclein exists as an unfolded monomer. Then when you start to get kind of toxic conditions, it can aggregate, and then it can build up into fibrils. And it's these fibrils that are the basis of the Lewy bodies, which are like the protein clumps that you see in the surviving neurons in Parkinson's disease brains. And these aggregates and these fibrils are thought to be quite toxic. And it's thought that these are the things that lead to the degeneration of the dopamine neurons in Parkinson's. So in my cells, I have wanted to measure the level of the unfolded alpha-synuclein monomer, as well as the level of the toxic alpha-synuclein aggregates. So when I measured the levels of the unfolded monomer in the E326K, there wasn't an increase in alpha-synuclein monomers, unlike what we see in the GD causing mutation L444P. However, when we measure the toxic alpha-synuclein aggregates, there is a massive increase in the levels of aggregates in E326K cells, which is similar to that of the GD causing L444P cells, which is really interesting because of the previous slides I've shown you, the E326K mutation seems to behave really differently to the L444P mutation, yet they're still both causing a lot of alpha-synuclein aggregation. So then it was kind of thinking, okay, what's going on in these cells? What is happening with this mutation to lead to this buildup of alpha-synuclein? Is it a reduced function of the lysosome? Is it being misfolded in the endoplasmic reticulum? Well, we know that's not the case because of the slides that I've just shown you. So I've turned my attention now to figure out whether something's going on in the cells to do with the levels of fats in the cells. And is this maybe potentially going to be involved with the mitochondria, which is something I want to look at in the future. So just a bit of background on fats in the cells. Normally in cells, in healthy cells, the levels of fats are really tightly regulated. And if you get changes in these levels of fats, it can become toxic, especially if you get increases of fatty acids. because these fatty acids can bind to different things in the cells and cause toxicity within the cell. So for a way for your cell to protect itself from these free fatty acids, they package them up into these little things called lipid droplets. So these lipid droplets can be released from the cells and broken down by neighboring cells, or they can be sent to the mitochondria to be used as an energy, a fuel source for them to create energy. However, if you get an increase that keeps increasing in fatty acids, this increase in lipid droplets, this is eventually toxic to the cells, and it may be this that is causing degeneration in Parkinson's disease. So I grew some of my cells on cover slips, and I stained them with a dye that stains for these lipid droplets. And when you look at them under the microscope, the lipid droplets are these bright green kind of circular structures where the white arrow is pointing to here. And you can see, I think, quite easily by just looking at the pictures that in the E326K cells, compared to control, compared to the other two Gaucher disease causing mutations, there is this huge increase in the number of lipid droplets. And that's the same as what we see when we quantify it. And we've done this with a lot of different images and quantified a lot of cells. So from this is suggesting that in the E326K, there may not be this loss of function, not be this endoplasmic reticulum stress, but potentially some alterations in the way that the cells deals with fats. Is there an increase in the number of fatty acids because there's a dysfunction in the mitochondria, so it's not breaking them down? Is there a dysfunction in the way that these cells can release the lipids to be broken down? Is there a change in the way that the mitochondria behaves? Do they use more fatty acids for fuel in these cells, so the cell is making more fatty acids and packaging them up more to be given to the mitochondria? So this is all something that I want to look at next. So the difficulty is with the stuff that I've done recently is it's just done in human skin cells and neuroblastoma cells. These aren't the cells that are dying in Parkinson's disease. So we want to see if the same thing is happening in Parkinson's disease affected cells. Is E326K still behaving differently to the other Gaucher disease causing mutations? Do we still see this increase in fats and lipid droplets? So what we've just started in the lab this week is started to make dopamine neurons, which are the cells that are affected in Parkinson's. And it's quite cool actually, because what we can do is we can take skin cells or blood from participants when you come in. We can then grow these in a dish in the lab, and then we can add different chemicals to them and reprogram these skin or blood cells into inducible stem cells. We can then add other chemicals to these stem cells and generate kind of different cell types. And what cell types that we will be generating will be dopamine neurons. And then in the lab, we actually have the cells that will be affected in Parkinson's from those people with Parkinson's, that we can look and see how these different proteins are working and what pathways are involved. So it's really cool and it's really interesting and quite exciting work to do. So that's what we're hoping to do in the coming months. But yeah, I just want to end with just saying thank you to everyone that comes in and that gives us your blood and your cells, because without all your participation, I wouldn't be able to do any of the work that I do in the lab. So yeah, thank you very much. I hope that was interesting. Just stop sharing my screen now, Naomi.
Thank you so much, Laura. That was excellent.
Thank you.
I'm now going to hand over to Elisa. So Dr. Elisa Menozzi is a neurologist and a current PhD student at UCL, and her current research focuses upon the relationship between the microbiome and the brain, and she will be discussing the PD biome study with you now.
Thank you so much, Selen, for the introduction. Can you just quickly confirm that you can see my slides?
Yeah, I can see your slides.
Lovely, perfect. Thank you, and of course, good morning, everyone. It's really a pleasure to be here today and to be able to present you these new and very exciting research projects that we are currently doing at University College London and Royal Free Hospital, which falls under our PD Frontline and RAPSODI study. And this project is particularly investigating the role of the microbiome gut-brain axis in Parkinson's disease. Now, just to give you a little bit of background about the gut microbiome and its relationship to Parkinson's disease. Let's start by defining what the gut microbiome is. So the gut microbiome is essentially the community of the bacteria that live in our gut. Just to give you a rough estimate, the number of bacterial cells that live inside of us is 10 times more than the total number of cells in our body. So it's a huge community. And these bacteria are extremely important in our health because since birth, they essentially build up our immune system, they protect us towards infection, they produce a lot of important vitamins for us. So they really, really are important for us. And the interesting thing about the microbiome is that it's mostly determined by what we call environmental factors. So food that we eat, lifestyle habits, medication, so all factors that we can potentially control. And the relationship between gut microbiome and Parkinson comes from several studies which have been investigating the gut microbiome composition in patients with Parkinson's disease, and have concluded that people with Parkinson seem to have a slightly different composition of the gut microbes compared to healthy individuals. And more interestingly, some years ago, it was shown in animal studies, especially in mice studies, that transplanting specific gut microbes in a mouse which is healthy can promote the progression to Parkinson's disease. So that mouse, which is healthy, after receiving microbes, develop Parkinson's disease. And this really, for the first time, opened up the possibility that these gut microbes might have an impact in the onset of Parkinson's disease. We have already talked about Parkinson and GBA, but just to very briefly summarize, we know that having a GBA mutation is a risk factor for the development of Parkinson's disease, but is not enough, because only a minority of people who have a GBA mutation will develop later on during their lifetime Parkinson's disease. And what we still don't know so far is what are the factors that together with GBA can promote the progression to Parkinson's disease in some individuals. So the question that we really want to address in this study is to try and identify whether a specific composition of gut microbes can increase the risk of development of Parkinson's disease in people who have a GBA mutation. In other words, we want to look whether maybe some bad microbes in a minority of individuals can lead to Parkinson's disease, or on the other hand, whether some good microbes in most of the individuals who carry a GBA mutation will actually prevent the development of the condition. And together with this, we also want to investigate what are the metabolites, or let's say the chemicals that these microbes produce. Because the gut microbes live in the gut, but they release chemicals that go all in all our body fluids. And of course, these chemicals have an impact on many, many functions. Now, to do this, we are currently enrolling participants from our PD Frontline and RAPSODI study and ask them to take part in this study. We are specifically recruiting 4 different groups of people. People who have a GBA mutation but have not manifested any signs of Parkinson, so carriers of a GBA mutation without Parkinson's disease, as well as people who have a GBA mutation and also have Parkinson's disease, people who have Parkinson but have tested negative for changes in the GBA gene. And within this group at the moment, we are particularly trying to enroll people who are drug naive, so people who have never started any medication for their Parkinson's disease. And the final group that we are enrolling in the study is composed by spouses of people with Parkinson's disease. And what happened with this study? So the first step is that we send you home a saliva and a stool collection kit to collect these samples. This is quite easy. The collection kits come with instructions provided, and you can collect at home the samples. Then we generally ask participants to return these samples when they come to the Royal Free, because the second part of the study involves a visit, a face-to-face evaluation at the Royal Free Hospital in London. And during this clinical evaluation, we ask you some more detailed questions about your medical background, the medication you might be on, as well as we ask you some questions about the diet, the lifestyle habits, the medication, your bowel movements, because as I mentioned earlier, all these factors are extremely important in determining the gut microbiome composition. When you come to Voyal Free, we also ask you to donate some blood, some urine, to some people also some skin. And you have heard from previous speakers, especially Laura, why this is so important for us. And we will also send in 1 year time another saliva and another stool collection kit at home, because we would like to see over time whether there is any change in the gut microbiome composition, as well as any change in the clinical features that you might have, to also try to find a correlation between changes in the gut microbiome and changes in your clinical condition. And what we are interested in looking at, as I mentioned earlier, is what are the microbes and what are the chemicals that are more or less common in people who have a GBA mutation. Because the final goal is try to understand whether specific microbes can predict the development of Parkinson's disease over time. And we also want to understand how factors such as diet, medication, lifestyle habits can ultimately influence the gut microbiome. Now why this is important? Well, because as you imagine, collecting a saliva and a stool collection and a stool sample is very easy and non-invasive. So potentially the assessment of the gut microbiome might become an important marker of Parkinson's disease risk in those people who carry GBA mutation, a marker which, as I said, is non-invasive and very easy to look at. And we know that the gut microbiome can be controlled and manipulated by us by a different diet or a different lifestyle habits. And as I always say to patients, the brain is very difficult to reach, but the gut is very easy to manipulate. So we perhaps in the future hope that we might be able to address those people who have a GBA mutation and maybe have a higher risk of developing Parkinson's to intervention that can change their microbiome, intervention that are easily to apply, and this intervention might prevent the development of the condition. And with this, I would end up here, but let me just take 30 seconds to, first of all, thank you again, all of you who are on the call, for your attention, for your participation. And since I have recognized some names that I know on the group of participants, people who have already taken part in the study and have attended Voyal Free, I would just like to take this opportunity to thank them again. Thank you for your support, for your enthusiasm in the project. For whoever of you has any interest in knowing more, having some more information, this is the email address, pdbiome@ucl.ac.uk. You can drop us a line, email us, and we will be more than happy to answer you. And of course, I'm happy to take questions at the end of the webinar. Thank you.
Thank you, Dr. Menozzi. That was brilliant. I will now present our final speaker, who is Professor Anthony Schapira. Professor Schapira is the principal investigator of both the PD Frontline and RAPSODI study. He is the head of the Department of Clinical and Movement Neurosciences at the Institute of Neurology and is a professor of neurology. Professor Schapira is an expert in Parkinson's disease and has a particular interest in the relationship of the disease to the GBA gene. His research includes the identification of new compounds that are candidates to slow Parkinson's progression, and he will now discuss the role of Ambroxol as one such compound.
Thank you very much indeed, Naomi. Everyone will be relieved that I don't have any slides. So I'm just going to really talk to you about Ambroxol, and to do that, I'm going to ask some simple questions and then try and answer them. So the first is, of course, what is Ambroxol? So Ambroxol is a cough linctus, and it's been around for about 40 to 50 years. And it's available over the counter, predominantly in Europe, but not actually in the UK or the United States. And it's been used over all of this time for simple upper respiratory tract problems. So what is its relevance to Parkinson's disease and GBA? Well, a number of years ago, people were looking to see, are there any compounds that actually can increase the activity of GCase? You'll remember that GCase is the enzyme, the active protein, that's produced by the GBA gene. And people that were interested in Gaucher disease asked the question, are there any drugs out there that can actually increase the enzyme activity and therefore correct the consequences of the GBA gene that causes Gaucher disease? And a group in Toronto looked at thousands of drugs that were currently available and being used, prescribed or sold, to see if any of them actually were able to do this. And Ambroxol came up as the top target. So how does Ambroxol work? Well, Laura very interestingly showed us that a mutation of the GBA gene, so when the gene is faulty, that mutation can cause a reduction in the enzyme activity and also cause it to misfold, so it looks different within the cell. What Ambroxol does is to bind to that protein and correct its formation, correct its folding, so that it then can transfer normally through the cell and go to that part of the cell, the lysosome, where it's needed to act. So Ambroxol is what we call a chaperone, and it helps increase the activity of the GCase enzyme. So this made it particularly relevant to Gaucher disease. But also, of course, we asked the question, and we started on this work a little over 10 years ago, is Ambroxol potentially of use in Parkinson's disease? So that actually brings me to the next question that I want to address is, what's the relevance of Ambroxol to Parkinson's disease? Well, we know that there is a very strong association between GBA mutations and Parkinson's disease. Although not everyone with a GBA mutation will develop Parkinson's disease, indeed, only a minority. But nevertheless, GBA gene changes are the most common association with Parkinson's disease. Now, by looking at the brains of those people who died with Parkinson's disease and donated them to the Queen Square Brain Bank, we found that those that had carried a GBA mutation during life and developed Parkinson's disease had a very low level of the enzyme activity in the brain. In a way, this is not particularly surprising because we know that GBA gene mutations do that. But what we found in addition to that was extremely interesting and exciting. We found that those brains from Parkinson people that had no GBA mutation also had a reduction in the GCase activity. Not as severe as in those with the mutation, but nevertheless still significant. So here we had both those with and those without Parkinson's disease-related GBA mutations who had a reduction in the enzyme activity. So a reduction in GBA activity is relevant not only to those patients with Parkinson's disease who carry a GBA mutation, but also those who don't. So with that in mind, we then began to look at the question, what is the evidence that Ambroxol may help people with Parkinson's disease? And the intention here was to slow progression, or potentially even in some, maybe to stop the development of Parkinson's disease. So we started with some very simple models. For instance, the skin cells that Laura talked to us about before. We took skin biopsies from people with Parkinson's disease, people with Parkinson's disease with GBA mutations, controls, and so on. And we found that in these cells, Ambroxol significantly increased the level of the enzyme activity. So this is very encouraging. We then, over the next few years, went on to look at a whole range of additional models, including stem cells that we've generated from patients. We've looked at flies into which we've put a GBA mutation. We've looked at fish into which we put a GBA mutation. And we've looked at mice into which we put different GBA mutations. In all of these models, we found that Ambroxol increased the activity of the GBA enzyme. And this was really exciting. But one other thing which we found from all of these models, that not only did Ambroxol increase the activity of the enzyme, it also reduced the level of the alpha-synuclein. You'll remember that the alpha-synuclein is the protein that accumulates in Parkinson's disease, whether or not people have a GBA mutation. So the observation that Ambroxol was capable of not just increasing the GBA enzyme, but also reducing the synuclein made it a really great candidate to see if it would work in people with Parkinson's disease. So that's when we turned to the first trial of Ambroxol, the so-called AIM PD trial, which is what we call a phase 2 study. And this was very generously supported by Cure PD. And the purpose of the AIM PD trial in a relatively small number of patients was to ask the question, is it safe in people with Parkinson's disease? Is it safe if you take it for 6 months? And does it get into the brain? And does it increase the GBA protein activity? So those are simple questions that needed to be asked by looking at this small patient group who took Ambroxol for 6 months. The results we published in 2020, and they showed that indeed, as expected, Ambroxol is perfectly safe. We have no significant complications with the use of Ambroxol, which speaks to its safety for many years, many decades as a cough lintus. It was very well tolerated by the patients. It had a little bit of a bitter taste, and in that study, people had to take quite a lot of tablets, although we've now gotten around that problem. But those were the only complications. We also found that by looking at the spinal fluid, that the Ambroxol was able to get into the brain in relatively high concentration, and most importantly, increase the GBA protein within the spinal fluid. So the trial was successful on all of those counts. Interestingly, those people in the trial also found that their Parkinson's disease seemed to be quite a bit better. But we were very cautious about the interpretation of that observation because this was not a placebo-controlled study. So with that information and all of the laboratory data that I've briefly summarized, we've now moved to the next phase of Ambroxol development, and that is a phase 3 study. So this is the gold standard to test a drug for use in people with a disease. And what we're hoping to do with Cure Parkinson and partners is to develop a phase 3 study that will look at probably around 330 people with Parkinson's disease, half of whom will have a GBA mutation, and the other half will not have a GBA mutation. Of these 330 people, they'll be divided randomly into those that will receive Ambroxol, 3 tablets a day or a placebo. And this is absolutely necessary for us to be able to understand the results at the end of the day. And the end of the day will be 2 years after starting the medication. So this, in a way, is a relatively short study in the greater scheme of things, although 2 years does sound a long time. But it will unequivocally show for us whether Ambroxol is capable of slowing down the progression of Parkinson's disease. We'll also be looking at lots of other things for people with Parkinson's disease and the use of Ambroxol, but that will be what we call the primary endpoint. So we're hoping to start this study later this year or at the very beginning of next year. And that is why RHAPSODI and PD Frontline are so important for people, because the gateway into this and other clinical trials that have been discussed this morning is through PD Frontline and RHAPSODI. So just to recap on the excellent presentations from Selen and Naomi, anyone with Parkinson's disease can join PD Frontline. People who know they have a GBA mutation or relatives can join RHAPSODI. If you're not sure about which one or other you should join, then just contact us and we'll let you know. But it's so important, not only for our research, but to offer people the opportunity to enter clinical trials that can alter the future of Parkinson's disease. So I'd like to add my thanks to the panel, and in particular, the thanks to around 150 people that have been participating this morning. And I hope that you found this morning interesting and helpful and informative. And I'll now hand over to Romy, who's going to take us on from here.
Hiya. Thank you to all of our panelists for their brilliant presentations. We'll now be hosting the Q&A section of the webinar. We won't be able to cover all questions today, but we'll cover as many as we can, time providing. So feel free to use the Q&A feature, which should be on the bottom right of your screen, to ask any questions to put to our presenters. I believe Selen has a couple of questions that she'd like to answer to begin with.
Thank you, Romy. Yes, so I've been receiving a couple of questions about the timeline that I presented. I know that Naomi has as well. I just wanted to clarify the timeline for everyone that's asking. So for those people that perhaps registered in 2020 and 2021, the timeline will be slightly extended due to the delays we experienced during COVID. And we will be in contact if your sample is not suitable for sequencing. Although I want to assure everyone that the samples are viable for up to 5 years in those saliva kits. So for those people that signed up in 2020 and 2021, due to the pandemic, we had to halt our sequencing process, which led to quite a backlog. We are working through this and we will contact you once we have your results. And we are very, very sorry for this delay. Thank you.
Yes, it's probably worth adding there, Selen, that the reason why we had to halt the process during the COVID pandemic was twofold. First of all, the transmission of saliva in the post, but secondly, all of the consumables, all of the plastics, the chemicals, etc., that we normally use in looking at the genes had to be diverted to the diagnosis of COVID. So there were none of these facilities available anywhere around the world other than to do COVID. So we're now catching up though, and catching up very quickly.
So another question for Selen and Naomi. How many subjects are already enrolled in RHAPSODI and PD Frontline, and what is the goal recruitment number?
So overall for RHAPSODI and PD Frontline, it's 4,000. PD Frontline currently has 1,880 participants. And I'll let Naomi answer the question for RHAPSODI.
Yes, thank you, Selen. So RHAPSODI currently has 930 participants, And yes, our overall goal between both studies is 4,000 participants.
Great. And a question for Dr. Menozzi. Can any family members volunteer for your research, or is it only the spouses of those with PD?
Thank you. That's a really good question. So we are mainly concentrating on spouses only because essentially, if we think about the gut microbiome and the factors that can influence it, as I said, diet, eating together are all factors that can influence. So we are recruiting people who live together, so households, and mainly spouses for this reason, and also because we are looking at people who are more or less of same age. So if you are a family member who lives with someone with Parkinson or someone who has a GBA mutation, you live together, you have more or less the same age, so absolutely you can be recruited in the study. Otherwise, this is becoming a bit more difficult because, of course, also the microbiome changes if you are in your 20s or 30s or in your 60s or 70s. So people of more or less same age who live together can absolutely participate as healthy controls.
And another question for Dr. Menozzi. Will you also look at fungi for the PD biome study?
Another good question. So for the moment, we are mainly looking at bacteria, only because the techniques that we use to look at bacteria or fungi are a bit different. But for the PD biome project, we are part of actually a bigger network. And the project is funded by the Michael J. Fox. So we are working with other groups also around the world, which are looking into this. And I'm not excluding that we might also do some sequencing in this regard in the future.
Another great question is whether there is a link between PD and type 2 diabetes. And this might be a great time to plug that Cure Parkinson's recently hosted a conference about this exact topic. So Helen Matthews might want to jump in here. But all the recordings are on their official website.
Yes, thank you. And I think that I probably need Tony to help me on this one, actually, given that I'm not a clinician. However, certainly we are seeing some correlation between insulin resistance and Parkinson's, and it's a very, very active area of research. I think people who have diabetes and Parkinson's definitely need a different treatment path. And I think our hope is that these diabetes treatments that we are testing will prove as standalone treatments for all types of Parkinson's, not just people with insulin resistance. Tony, you may want to add some additional clinical insight.
Thanks, Helen. Yes, well, you're absolutely right, of course. And there is an epidemiological link between type 2 diabetes and Parkinson's disease. So it appears that type 2 diabetes does increase the risk or is associated with an increased risk of Parkinson's disease. And what we're interested in is it the diabetes that promotes the Parkinson's disease, or is it something else which helps to increase the risk of both the Parkinson's disease and the diabetes? But a very active area of research, and as you say, translated now into using some of the diabetes therapies in Parkinson's disease.
And another question for Professor Schapira. Could you please elaborate on the effect that Ambroxol has had on people with Parkinson's in phase 2 trials? How were their symptoms specifically affected?
Well, their motor symptoms are improved. People notice an improvement in tremor, and so on. So overall, the measure of Parkinson's disease, and we can do that using something called the UPDRS, improved significantly. But as I said, our interpretation of that was cautious. It clearly was very helpful in that, first of all, it meant that the Ambroxol was not interfering with any of the normal Parkinson drugs. But because this was what we call an open label study, not a placebo-controlled study, it was difficult for us to say that this benefit was definitely related to Ambroxol, but nevertheless encouraging. And naturally, That's why we're doing the phase 3 study.
And on the same vein, what dose of Ambroxol is to be used in phase 3 study, do we know?
Yeah, we're planning to use the same dose as we used in the first study, so 1,260 milligrams, but will now be available in a much more convenient form of 1 tablet 3 times a day.
And can you be on the Ambroxol trial whilst on gut microbiome research? Will this offered automatically if eligible?
Yes, absolutely. I mean, I would imagine that the best gateway is to join up to RAPSODI or PD Frontline and to come through the microbiome study and then into the next Ambroxol study. That would be a natural course of action for all those people that are eligible. And of course, those people that will be in the Ambroxol study will have an opportunity to participate in other areas of related research in the clinical trial itself. So yes is the answer, but probably the best way through that is Frontline and RAPSODI, because we would need to know whether the person has a GBA gene change or not.
If you're an older PD sufferer, do these trials still apply?
So the inclusion-exclusion criteria, as they're referred to, will include anybody up to age 80, but they will have to, or 75 rather, they will have to have certain aspects of their Parkinson's disease that fits with the criteria that we're looking at. So age is not a bar, but other aspects of Parkinson's disease will be looking at more carefully.
And are there any diet and stress factors related to the GBA mutation?
Do you want me to answer that?
Yeah.
Or maybe I'll hand that over to Elisa, as it includes the diet, because she's helping to study this.
Well, I think the answer is that we still don't know, and we are looking into this. We are looking whether the GBA mutation itself can have somehow an impact on the gut microbiome, because we don't know. As I mentioned earlier, most of the variation in the gut microbiome is due to environmental factors, so not our host genotype. But we're looking into this because potentially this could also influence it. And then by asking participants which type of diet they're on, when we will look at their microbiome, we will be able to see whether there is any association between specific diets and specific composition of the microbiome. This is an open question, which I think is very important to answer. And maybe in the next webinar, I'll give you a more definite answer.
That's great. And could you advise whether there are any specific microbes, bacteria that are absent or could be introduced to people with Parkinson's to reduce PD symptoms, i.e. like probiotics?
Well, this is another good question. So for the moment, the use of probiotics have been applied in 2 randomized clinical trials, looking at improvement in constipation as an outcome. And both of them have been successful in treating constipation. We still don't know, just because it has never been really studied in clinical trials, what is the effect on probiotics, for example, on motor function or other symptoms of Parkinson's disease. For the moment, there is also no, let's say, clear definition on which type of bacterial strains or bacterial species can help more in Parkinson. What we do know is, for example, that increasing the intake of dietary fibers, so fruit, vegetables, definitely improve bowel movements and motor function in Parkinson's disease. But we need to do more studies to look at specific bacterial species in Parkinson's disease, and of course in Parkinson's disease with GBA, which has never been investigated. So we will be looking into this.
going to hand over to Derralynn Hughes.
Hi, thanks. I just wanted to offer perhaps a tantalising insight here from the world of Gaucher disease. So it's always been a somewhat of a puzzle to us as to why Gaucher is actually so common, and particularly amongst the Ashkenazi population. And so there's a suggestion that being a carrier of GBA actually over many generations, many thousands of years, provides an advantage for those people. There's recent work suggesting that those storage materials, those GBA-related fats that were described to you, particularly the glucosyl sphingosine, might actually be effective in preventing certain sorts of infections, so in helping the body to fight against particularly TB, but probably some other infections. And in fact, glucosyl sphingosine is likely to be a sort of antiseptic within the cell. So it may be that there is some benefit in pursuing those links, because what may be beneficial for preventing infections that were really important over the evolution of humankind may actually be a disadvantage in terms of the microbiome. So it's just a thought, but maybe worth having a bit of a discussion about.
And a final question, are there any limitations on participation of multiple trials at one time? I'll put that to Professor Schapira.
Yes, I think there would be in terms of intervention studies. So it's almost always the case that if a drug is being tested in a clinical trial, participants are not able to be in another clinical trial investigating a different drug at the same time. But people in an intervention study, such as, for instance, Ambroxol, can participate in other types of research that doesn't involve the taking of a different compound, which might complicate the interpretation of the Ambroxol results. But they can, for instance, participate in the microbiome project.
Thank you, that's brilliant. Thank you all again, and thank you very much for joining us today. I'll now hand back to Selen and Naomi for any closing messages.
Thank you so much, Romy. Thank you to all our panelists and all our participants. I just want to apologise again for the technical difficulties and the delay in starting. I'll hand over to Naomi to give the closing remarks.
Yes, thank you, Selen. We just wanted to say a very big thank you again to all our panelists who gave us some very insightful discussions, and to all our attendees and participants, because without your help, we wouldn't be able to do any of our research. So thank you so much. Your contributions are truly invaluable to our work. And if you have any questions at all, please don't hesitate to get in contact with either Selen or I. Yes, thank you.
I also wanted to add, this will be recorded and available to watch later if you have any questions or if you weren't able to take notes at the time.
Thank you.
Thank you.
And have a lovely day, everyone.
Thank you very much. Bye.
Bye.